Hi everyone. Just want to say thank you for taking the time out of your day to sit and listen to my research, and I hope you enjoy it. So you might already know this, but it's on relationships between waterfowl beak morphology, ecology, or feeding ecology, and sensory brain region pathway structure. It's pathway structure and brain region structure. Fumbled over my words a little there. And so, a little brief introduction. So waterfowl are made up of a wide variety of species, including ducks, geese, and swans. They exploit a wide variety of food sources, such as grasses, aquatic plants, fish, insects, amphibians, worms, mollusks, and many more. There's very large abundance, and that paves way to various feeding ecologies or feeding strategies. And based on this, past researchers have done the hard work for us and classified them into 4 groups: dabblers, divers, active pursuers, and grazers. And some of these classification exemplars: for a dabbler, we have a mallard, and dabblers pluck at vegetation and immerse their beak in water to filter small seeds out. They can also use their tongue to create a pumping action, trapping prey in the lamellae of their beaks. And then we have an active pursuer, such as the red-breasted merganser. And how they feed is they have more of a narrow serrated beak that allows them to catch fast-moving agile prey. And then a diver, a ring-necked duck. And divers have more of a broad flat beak that allows them to pick up plant tubers and sift through sediment. And lastly, the grazer, or the Canada goose in this case, eat more vegetation, so they usually have more of a short, stout, and powerful beak to grab tubers from the ground. And so the mechanism behind the tactile sense is the sensory receptor cells, or mechanoreceptors, that take in information, touch information, and then they conduct out along the trigeminal nerve, which is a focus or focal point, and then it terminates in the principal sensory trigeminal nucleus, PRV, and the nucleus basorostralis. Now I'll mainly be focusing on the PRV I have focused on the PRV. I haven't focused on the nucleus basorostralis. That's for the future. And so for the significance, all this information, PRV cell counts is one thing that I've gathered. Trigeminal nerve fibers counts, mechanoreceptor counts. We calculate the mechanoreceptors by counting pits that are in the beak, and the pits actually house the mechanoreceptors. So in the end, we use CT scans, and we were able to quantify that. It was more of a what you could say, exhaustive technique of doing it, but grateful that we had help with that. And so in the end, this is going to give us a better understanding of evolutionary mechanisms of sensory specialization and its relationship to phylogeny, ecology, and allometry. And lastly, I will give you a little sneak peek at my poster. And one thing that I want to mention in going in is to keep an eye on the keys, because they're very important for understanding and interpreting the graphs. I hope you enjoy it, and I hope to meet you soon. If not, I hope you enjoyed the information you received here, and you have a great day. Thank you. Goodbye.